So we're going to start today's video off by going through the questions from our last class, just briefly. So number one, why do more complex multicellular animals require a circulatory system? Well, the problem is if you have multiple cells, okay, you have very low surface area. Okay, so this is your total surface area here, okay, compared to one cell, we have all this surface area, okay, so what you want to say is you have a low surface area to volume ratio, okay, meaning that essentially not all the cells can have their needs met. Okay, they don't, if you think this cell here, it does not have ready access, or it can't easily access the nutrients or whatever is out here. Okay, so we need a better way to deliver, or a more efficient way to deliver those nutrients, take away waste, all that stuff for those cells. Functions of the circulatory system, that there was a list there in the slides, including things about oxygen and carbon dioxide, hormone transport, etc. You can look through the slides of the book if you need to find those. Three features common to all circulatory systems, that would be the fluid, the pump, and the tubes that contains the fluid. Graphic organizer I won't go through. Why can insects and other invertebrates survive? Well, they have a very low basal metabolic rate, so they do not require a rapid flow or rapid delivery of oxygen and nutrients, because they don't really do respiration as much. Okay, so they do not need to be incredibly efficient. Okay, they can get away with being, a bit less efficient. Okay, explain the difference between the pulmonary and systemic. I'm sorry I did not address this in our class. We'll look at it more when we look at the heart specifically, but since it's here, pulmonary means lungs. So the pulmonary circuit, if this is your heart, okay, we know that it goes to the lungs, and then back to your heart. Okay, that's called the pulmonary circuit. The systemic circuit is when it goes from your heart to your body and then back to your heart. Okay, so pulmonary goes to the lungs, systemic goes to the rest of the body. Structural feature of the heart, again, sorry for not addressing this. Hopefully you found it in the book, but it should be the dividing wall between the two parts of the heart, called the septum. Okay, or a wall. What this does is it stops the blood from each of these sides mixing. I'm oversimplifying it here, but this blood stays separate to that blood. Okay, so when this part contracts, okay, the blood only goes out here. And likewise, when this part of the heart contracts, the blood only goes down there. So it keeps your two circuits separate. Lastly, how does the digestive system contribute to the function of the circulatory system? Well, your heart is a muscle which contracts all the time, which means it needs needs glucose. It does need oxygen as well, but in terms of digestive system, it needs glucose. So it needs the digestive system to provide those things to the heart itself so that it can pump. In addition, all the structural parts, so the things the tubes are made of, the fluid itself, all of that stuff is made from the things we get in our food. Okay, so all of that is provided from the digestive system. Okay, as always, if you have any questions about those, please shoot me an email. Today's class we're going to be looking at blood. So this is the fluid, right, inside our circulatory systems. Our objectives, we'll come back to this at the end, but we're going to look at exactly what blood is and what each of those parts do. We're going to take a look at blood typing, and then look at some disorders and technologies related to blood. So back in the day, a few hundred years ago, they used to think, so this is incorrect, do not learn this. they used to think that there were these things called humors, so like 4 types of liquids or elements, and that you needed to maintain balance of those in your body. And so, if you were sick, what they thought was that there was an imbalance in these humors. So that would mean you needed to take out some of your humors, okay, to restore the balance. So what this led to, if you notice here at the bottom, blood is one of the humors in the bottom left here with air. And so what this led to was, if you're sick, we need to balance out your humors. Let's take out some of your blood. So for a long time, this practice of bloodletting was performed. People actually did die from it. They would, what they would do is they would cut, as you can see here, a hole in you near a vein, and they'd allow the blood to drain out, hoping that it would balance out your humors, okay, thus causing you to be healthy again. This is a diagram from an early manuscript showing the areas in which you would need to bleed people from. I'm not sure if they thought there was a difference between where you bled people from, but nevertheless, you can see that they had quite a lot to say on it. How much scientific evidence they had, I'm not sure. So, what we need to know about blood. In terms of tissue types, we call it a connective tissue. Other tissue types involve muscle tissue, nervous tissue. I forget the other ones, but connective tissue is kind of like your bone. Okay, that was another type of connective tissue. So the definition of a connective tissue is a cell or cells suspended in an intercellular matrix. So you have cells and there's stuff between the cells. Okay, intercellular, between cells. The average person has about 4 to 5 liters of blood, and you can remove about 15% of this without any real compromise in function. So you can still be quite healthy if you lose about up to 15% of your blood. Obviously, if you're sick, or you're elderly, or you're very young, this may be different. But this is why we are able to give blood, because you actually can survive without all of it for periods of time. What is in blood? Well, like we said, we have the cells in an intercellular matrix. So the plasma is the liquid part of the blood. And we could say that, so I'll write down liquid here. This is the intercellular matrix. It's the stuff between the cells. So then what we have is the cellular parts. Cells. And those are your red blood cells. So RBC is red blood cell. White blood cell. And your platelets. Okay, platelets, think of them more as parts of cells. They're not actually a cell in themselves. So this yellowy goopy stuff is the plasma. This is the stuff where when you scrape your leg and you have that goop coming out. That's very similar to plasma. It's actually the tissue fluid, but it's very similar to plasma. So plasma is a watery substance with a whole bunch of proteins inside of it. Okay, in addition, anything that's dissolved in there, so looking at the bottom here, the ions, so salts that are in your blood, the glucose from your food, the minerals, and the gases, that oxygen, carbon dioxide, mostly carbon dioxide, not much oxygen. All that stuff dissolves into the plasma, because the plasma is the watery part. Okay, as you can see, it's mostly water. The proteins, you don't need to know what these do, but it would help if you remembered that you did have proteins inside that liquid. So when we're talking about DNA, we were saying how proteins are so important to your body. They are what do all of these many functions that your body does. This is just a short list of 3 proteins and their functions. Okay, so if you didn't have those proteins, you would be in a lot of trouble. Okay, a diagram showing you, so this yellowy part is the plasma. And as you can see, we have different cellular components. So let's take a look at one of them here. These are your red blood cells under an electron microscope. Okay, notice their structure, because we'll be addressing that. So what we call them is a biconcave, so 2 concave, hopefully you remember from if you know, when you have lenses, a concave lens is one that looks like that. Okay, cave goes in. What that does is it increases the surface area to volume ratio of your cell. So by doing that, okay, we have all of this surface area here. Okay, if we just had, you know, a normal looking cell that was round, okay, we'd still have a lot of surface area. Okay, but this takes up a lot more space. Okay, this one is much larger, has a much larger volume than this one. Okay, for the same amount of surface area. Okay, so by having this biconcave disc, we can actually fit in about 20 to 30% more surface area. Meaning, if you remember what's on the surface of these red blood cells or erythrocytes, is we have all this hemoglobin. So the more surface area we can get, the more oxygen we can carry. Now, your red blood cells are extremely small. They are one of the smallest cells in your body. They don't even have a nucleus, so what we're doing there is we're saving space. Okay, we're removing things that don't have to be there. As a result, red blood cells don't live for very long, but they perform their function with their hemoglobin of carrying oxygen. And that's the main function you'll need to know. The amount of red blood cells you have is regulated by erythropoietin. Okay, erythro from erythrocytes. Okay, so that's the red blood cells. And so we mentioned before how, you know, when athletes train at high altitudes, they produce more red blood cells. That's because it stimulates this thing called erythropoietin, which triggers this this process. Okay, so your red blood cells, you should know they are formed in your bone marrow. Okay, then they live for about 120 days. They go around, they do their stuff, and then they may then get, you know, once they've done their job, they'll get broken down in your spleen and they'll get recycled. Okay, 120 days is actually longer than I thought. Apologies for that. So then we'll break them down, recycle the good stuff, and then make more red blood cells. Next, we're going to look at your white blood cells. So that's this guy here. We will not go into much detail for your white blood cells. Okay, we will cover the main types, but you won't need to know much about each different type. Okay, so the main difference is that all of these guys have nuclei. Okay, compare them to red blood cells, which do not. That is the probably the main difference. Okay, in terms of their structure, they are the first. Actually, they're not the first line of defense, but they are one of the lines of defense against foreign particles. Okay, so these guys have the job of keeping your body safe from bacteria and other things like that, viruses and so on. Two main categories. One, we have granular leukocytes, and they contain harmful chemicals, so they can harmful to foreign cells. So they can release those granules, okay, release those chemicals and kill those harmful cells. And our agranular leukocytes, they swallow, okay, they eat the foreign cells and break them down inside them. Okay, just another representation of them. You won't need to know what these guys are necessarily. I would say know these names. So if I said, for example, an eosinophil is a type of what cell, you should be able to say it's a type of white blood cell. Okay. Next, we'll look at your platelets. So, these guys are, like I said, they're very, very small. You can see them in the diagram. They are smaller than a red blood cell even. They're not really cells. They are parts of cells. And what they do is they regulate clotting. So these platelets are what allow your blood to clot. Okay, you should know this word coagulation as well. That's a scientific term for clotting. So every time you have a scab, you cut yourself, you have a scab, that is through the platelets. Okay, that's what allows you to clot, which stops you from bleeding. This general process you should be aware of. If you remember that the platelets are what plug the gap, okay, and then you have fibrin. So platelets and then fibrin kind of form this mesh, okay, sealing the wall of the blood vessel. Okay, and obviously this takes it starts from minutes after you've been cut, okay, but then this healing process, depending on how wide the cut is, can take several days. Okay, but this will happen in a matter of minutes. One problem that platelets can cause is this condition of DVT. You may know that when you fly in a plane for a long period of time, they recommend that every couple of hours you get up, you walk around, you stand up, you stretch, you flex the muscles in your leg. What that's trying to do is push the blood around your body. What can happen is, if you've been sitting still for a long period of time, is that the blood near the bottom of your legs here can pool, so it can just sit around. And these platelets, your blood doesn't move as much, and so the platelets start to get sticky. Okay, because they start to get concentrated, they start to mash up against the walls here, and they can actually start to form a clot even when there's no cut. Okay, so deep veins, so inside your veins, thrombosis. Thrombocyte is another word for platelet. You don't really need to know it, but that's where that thrombosis comes from. And if this clot forms and part of it breaks off, this is now what we call an embolus. What that can do is, well, it's fine in this vein, but if that then goes into a blood vessel, let's say this blood vessel is this big, this embolus can actually clog that blood vessel, meaning that no blood is getting through. And if this goes to your brain or to your heart, that is what can cause a stroke or a heart attack. So this is one of the causes. Another problem related to platelets is when your body does not clot properly. Okay, so I don't know if this is a problem with the platelets themselves not producing enough platelets, but regardless of the cause, this is called hemophilia. Okay, and this is where your blood does not clot, okay, at all, okay, or to a limited extent. What this means is that every time you get a bruise, okay, a bruise is caused by small blood vessels bursting. Okay, that's why it becomes a bluey color. That's the blood kind of breaking down inside your tissues. And so, if you are hemophiliac, okay, if you're a hemophiliac, you get a bruise, you do not kind of stop bruising. All that bleeding continues. So this can actually be quite deadly. Okay, if it's happening in your neck, it means you probably have a major blood vessel that's not clotting. It's going to cause problems. It'll compromise your airway. But anywhere really in your body, look at these other ones, so I'm drawing all over this, that can be a major problem. Okay, because it's not going to stop by itself. So in each of these cases, they need to give you an injection of clotting factor. So something to artificially or to stimulate your body to clot. Okay, hopefully you'll be doing an activity on this later on, but how scientists go about figuring out if you have a good blood count or a high count. If you're looking for hemophilia, you'd probably expect fewer platelets. If you're looking for anemia, you might be seeing fewer red blood cells. If you're looking for things like leukemia, you may be looking for a high number of leukocytes. So counting blood cells is really important. It can be used to diagnose many conditions. So they have this thing called a hemocytometer. Hemo meaning blood, cytometer meaning cells, and cyto meaning cells, meter meaning counting or measuring. So what it is, it's basically a slide, okay, like we've used in class, except it's a little bit fatter, and it has this little grid on it. Okay, I think these, depending on where you are, might be about 1 millimeter, maybe. Sorry, no, they must be less than a millimeter. I think this thing is 1 millimeter. I'll need to look that up for you guys, but it's very, very small. And you put a small drop of blood on there, okay, and then you put your cover slip on, and it spreads the whole blood out. And so when you're looking down the microscope, you count how many cells you see, and so we can go, okay, in counting the cells that are within or touch the line, so we go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, so on and so on, and then we know this space, and we'll actually know this volume. Okay, it's not just a flat surface. We actually, this is slightly sunken as well. So we know the volume of this chamber. We know how many cells are within, let's say 0.1. Let's make up a number. Let's say we have 50 cells in 0.01 mils. We can then figure out how many cells you have per mil or per liter in your body. Okay, and if you fit within a certain range, if you're outside of a certain range, you may be diagnosed with one of these conditions. Okay, so this is a slide to remind you what you're meant to be doing. But hopefully now what you'll be doing is, or maybe after this video, you'll be performing a short activity where you will be counting these cells and using that to determine if someone has a certain condition or not. Hopefully you'll be doing that if I get around to it. Last thing we're gonna, sorry, not the last thing, but one of the things we're gonna look at now is blood types. You should know that there are 4 different blood types, and then we can have positives and negatives of each type. So I believe I covered this in class earlier, but what your blood type refers to is these things called antigens. An antigen is like a flag, and it tells your body which team these cells are on. Okay, they can be friendly flags, and they can be, anything else. Okay, so there's friendly and everything else. So if you are of blood type A, it means that your cells are showing antigen A or flag A. Okay, so it recognizes anything with antigen A as its own cells, so it's friendly. What that means is, and if your body's antigen B, your blood type B, it means you're making flag B. Okay, so your body recognizes flag B as friendly. If you are AB, you are producing both flags, and so both flags A and B are friendly. And if you are blood type O, you are not showing any flags. So the only things carrying flags are foreign cells or enemies. What this means is, well, your body wants to fight anything that is not friendly. Okay, so if you are blood type A, flag A is friendly. So you want to make things called antibodies. Okay, antibodies fight and kill other cells. So you want antibodies against B. Okay, because B is not your flag. Similarly, if you are of blood type B over here, flag A is not your flag. Flag A is the other team. So you want to make things to kill those cells. Remember, the reason you have antibodies is because most of the time, foreign cells in your body would be bacteria, okay, or viruses would be showing these antigens. Naturally, it would be very rare for you to have someone else's cells in your body. Okay, that's not a normal circumstance. So this is actually a helpful thing, okay, that your body can recognize and fight off foreign cells. If you are blood type AB, okay, flag A or flag B are friendly, so you actually don't make any antibodies against these flags, okay, because it'd be your flags. What this means is that even a cell with flag A on it, so let's say the blue one here, right, blue circle, a cell with this flag will also be friendly because it's showing flag A. Likewise, a cell that's only showing flag B, let's do this cell would also be recognized as friendly showing flag B. Or a cell showing both flags would also be friendly. Opposite to that, so blood type A, B, and O are kind of like opposites. Blood type O, the friendly team, has no flags. So anything with a flag is an enemy, so we make antibodies for both flags. Okay. What this means is that there's only certain blood types that can be given to or received from people. Okay. If you are blood type AB, okay, you are not making, you have no antibodies for these blood types, meaning that you can receive cells with flags on them, right? You can receive blood type A, so can receive A, because they show A flags and you have no antibodies, so showing flags is okay. You can also receive B. I should actually work from the left, so let me go there now. If you are blood type A, you can either receive from blood type A, because they're showing friendly flags, or you can receive from blood type O, because they have no flags. Okay, remember you have anti-B antibodies, so if you get blood type B, you'll kill those cells. If you get blood type AB, the A flags will be okay, but the B flags will be recognized and killed. Okay, but this is still every cell. So you can receive from A and O. If you're blood type B, you can receive from B, and again also O, because you have antibodies for the A blood type. And if you're blood type O, you have antibodies for all flags, so you can actually only receive from other people with blood type O. Okay, because of this, because of this we call blood type AB, I didn't finish off blood type AB. They can receive from AB, AB, and also O, because remember the O cells have no flags. Okay, so that's still okay. So what this means is blood type AB is called the universal receiver. Where can I write that? AB, universal receiver. And blood type O, okay, you'll notice that each of these blood types can receive from blood type O. So they are called the universal donor. Okay, because they can give blood to anyone. The next thing we need to worry about with blood types is this thing called rhesus factor. You would know this as the plus, or the positive or the negative when we talk about blood types. So the A, B, AB or O is the ABO blood typing, A, B and O blood typing, and this is the rhesus factor. Okay, so A positive means you're blood type A and you're positive for rhesus. I think it's called rhesus because it was first discovered in rhesus monkeys. Exactly how this works, the specifics I'm not sure of, but what we need to know is that it functions in a similar way to A, B, and O. Okay, the positive indicates a flag. It means you have the flag. If you have the flag is friendly. Okay, but if you have the flag, it means that if you are positive, it means that your cells are showing the flag. So you see that as friendly. So that's let me put that under positive here. Get rid of these. If you are negative, you do not have any flag. However, it means that if you are negative, you will make antibodies. For that flag. And if you are positive, you do not have any antibodies for that flag. Okay? So let me just get rid of that. So if you are, this is where the problem comes in, okay? If you are a rhesus negative mother, Okay, it means that your cells, no flag. Okay, they do not have the flag, but it means you have antibodies. And that's a problem if you are pregnant with a baby that is rhesus positive. Because you have these antibodies in your blood. So I'm going to do this little wise. You have antibodies in your blood for the flag. And your baby has, the cells of your baby have those flags. So what happens is, normally the blood of the fetus and the mother are actually kept separate. They don't really interact much. But during delivery, the mother can be exposed to that blood. Okay, it can enter the mother's blood. What that means is when she's pregnant the first time, these antibodies actually don't exist. Why that is because your body needs to first be trained. Okay, so before you've seen it, your body hasn't seen that flag before, so it doesn't actually have the antibodies ready. But once they have the first child, their body has seen the flags, and so now you see all these antibodies present here. And so now the body is trained to recognize that flag and kill those cells. And so if the woman now has another rhesus positive fetus, then the body's ready for it. Okay, these antibodies will then go and attack the cells of the fetus, okay, causing damage to the blood cells. Okay, and that can be a major problem for the fetus. Whether it can cause death or not, I'm not entirely sure. Okay, maybe look that one up. One of the technologies we'll look at just briefly is artificial blood. So this is kind of like the dream. At the moment, it does not yet exist. Okay, but the dream of artificial blood, because we have so many people that need blood delivered. Okay, especially during operations where they're losing a lot of blood. People who are suffering from a car crash or some other form of accident where they've suffered major blood loss. We need to replace that blood. Okay, because if there's no blood, we can't deliver oxygen. So the dream is to have artificial blood that will not induce any antibody reaction. Okay, we don't even want to have to care about what blood type you are. We just want to be able to give you this blood. Something that's not infectious, that can still transport O2, and that can sit around for a long time. When you donate blood, it only has a shelf life, I want to say it's a few months, maybe even less. Okay, but it can't last. Okay, for artificial blood, we want something that can keep going for a long, years even. Oh, I actually don't know what the outcome of this was. This was when I made the slideshow 2 years ago. This was more recent. I wonder how this went. I'll look that up and get back to you. Okay, but with stem cells, we'd have a way of making blood cells in a lab. Okay, we could just clone them, make many, many, many of them, and then we could deliver them to people when needed. In addition, if they're from your stem cells, okay, then these cells would essentially be yours, and then there would be no reaction in terms of antibodies. Okay, to end today's class, sorry, I should put the objectives back up. I'll be able to find those now, but hopefully you'll be able to do those objectives. Maybe flip back to the video, to the beginning of the video. To do so, I'd like you to answer questions. Let's not. So, 1 you can do. 2, good. That one. What I would not do is this one. Sorry, these 2 you don't need to do. Okay, so 1 through to 8 complete, and also hopefully 1 or 2 worksheets that I've managed to get to you. Okay, as usual, if you have any questions, shoot me an email or write on comment on this video in Classroom, and I'll do my best to answer it. Okay, thank you.